Hello students. In my last video, I explained that we have five major kingdoms of living organisms. That we have the kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. So when I discuss what is called prokaryotic cells and eukaryotic cells. So if you have missed that video, make sure you watch the video before this one. Then in this video now, I'm going to focus on the first kingdoms of living organisms. That's kingdom Monera. So this kingdom minerals contains the organisms that are prokaryotes. So, and I've already explained what prokaryotic organisms are. So these are organisms that have prokaryotic cells, the cells that do not have membrane-bound organelles. Now, these kingdoms subdivide into two major phyla. So the first phylum we have in these kingdoms, we call it phylum Schizophyta. Phylum Schizophyta. Schizophyta. And the phylum Cyanophyta. Now, the phylum Schizophyta and the phylum Cyanophyta, there are two major phyla of the kingdom Monera. So we call them Monerans. Monerans with N at the back. Monerans. So, and what are Schizophytas? So these are bacteria. Call them bacteria. Bacterial cell. And what are cyanophytes? So, or the cyanophytes. So we call them blue-green algae. Now, so the pseudophytes are the bacterial cells or the bacteria generally, and the cyanophyta or the cyanophytes are the blue-green algae. Now, there are general features that are shared by these two phyla, and one of it is that all monomers generally, they are prokaryotes, so that's no longer new. Then the second thing is that they are unicellular organisms, meaning that they are a single cell. Remember, um, cell, some organisms are made of a single cell, and this organism we call them unicellular, single cell organisms. So these organisms includes bacterial cells, amoeba, paramecium, glena, etc. like that. While some other organisms are made up of multiple cells, so like complex organisms. So for instance, now the animals, plants, fungi. So these organisms are made of multiple cells. So call them multicellular organisms. Multicellular organisms. Now, so bacterial cells, blue-green algae, they are unicellular organisms, meaning that they are made of a single cell. A single cell. Now, to understand this kingdom very well, because the target of this class is that I'm going to tell you everything you need to pass any exams under this concept. So take a pen and start to write down. So now the thing is that let's start with the first phylum of the kingdom Monera, and that's phylum Schizophyta. Schizophyta. Now, in this phylum, what happened is that you must learn to draw a bacterial cell of hand. So because it's really very important, because you may be given a structure in your exam and ask you to label the structure or tell the function of the structure. So in biology class, we deal with structure and structure related functions. So you must be able to draw a structure and relate the feature of the structure to the function. Now, let's start from this. So let's draw a bacterial cell and use that to explain the structure, the function of the bacterial cell. Now, so the function of each of the structures in the bacterial cell. Okay, please pay close attention to the diagram. So now I'm having three layers. Don't mind my diagram anyway. Okay, this is the third one. Then let's draw the nucleoid. So the nucleoid. Then they put the ribosomes. Now, so this guy, then um let's put the pili there. So now, so let's label this structure. So this is the structure of a bacterial cell. A bacterial cell. Now, the first thing you have to know is that this guy you have it is called the flagellum. Flagellum. So, and the function of flagellum is the organ of movement. Now, there's two things I want to tell you now. The first thing is that there are three organelles of movement that are actually found in organisms that are single cell. One of them is flagellum, and this is found in organisms like bacterial cell, euglena, sperm cells, etc., and also chlamydomonas. Now, the next one is cilium. Cilium is found in paramecium. So, and some larva of plasmodium like oocytes. So this is found in the mosquito stomach. Now, the next one is the pseudopodium. So this is generally found in amoeba cells. So most of these three organelles of movement in single cell organism. Now, the next structure here that's also very important is the pili. All this um all these tiny hair like, so they're called pili. And the function of pili is that since you know that bacteria are parasites, so they're also pathogens as well. So some of them are pathogens. Now they're pathogenic. Now the bacterial cells use pili to attach to the surface of the host cell or the host organisms. So this that function of pili. So pili is about attachments to the surface of the host cell. Since they are parasites, so they have to cause some infections and they have to attach the surface of the host cell. So that is the function of pili. Then the next structure here is the first layers of these boundaries. The outermost one is called the capsule. Capsule. And the function of capsule is very kind of dramatic because you know, let's say for instance now in the bloodstream we have the white blood cells that prevent our body from invaders or from intruders of now intruder pathogens. Now the capsule function in bacterial cell is to prevent being ingested, being eaten, being engulfed by white blood cell, for instance, or by some other cells. Now, because the capsule is protecting bacterial cells from being ingested by phagocytosis. Now, please listen very well. Phagocytosis. Now, I'll use this to explain some things to you, so pay close attention to it. Now, the first thing is that, remember I said the capsule prevents bacterial cells from being ingested, from being eaten, from being engulfed by bacteria, by white blood cell, or by some other cells that can engulf bacterial cells. So, the process of ingesting a cell is called phagocytosis. Now, what does this word mean? Phagocytosis. Now, listen very well. There are two ways in which something can go in and out of the cell. If something go into the cell, if something go into the cell, we call it endocytosis. When large materials go into the cell through the cell membrane, we call it endocytosis. When large materials come out of the cell into the environment, we call it exocytosis. Exocytosis. Now, if the thing that is going into the cell is a solid particle like another cell or food particles or some other particles that are solid, so that's when we call it phagocytosis. So therefore, solid particle going into the cell is called phagocytosis. And if that thing that is going into the cell is liquid, if that thing is liquid, we call it pinocytosis or pinocytosis. So now, pinocytosis, that's for liquid. So let me recap a bit. Now, so the first thing is that the bacterial cells have capsule which prevents them from being ingested by phagocytosis. So the meaning is that they prevent the protect bacterial cell from being eaten up by another cell. And remember, I said the word phagocytosis means that a cell is eating up another cell. Is that okay? So therefore now, if white blood cell eats up bacterial cell, so we call that phagocytosis. Because bacterial cell is a solid particle, so going into the cell of the white blood cell. So that's phagocytosis. So if something comes out of the cell to the environment, we call it exocytosis. Now, if something go into the cell and that thing is liquid, so we call it pinocytosis. So those are the terms you have to pay attention to. Now, the next thing we're talking about now is the cytoplasm. But before that, let's talk about the second membrane we have here. So the next layer from the outside, the middle one is the cell wall. The cell wall of the bacterial cell. The cell wall of the bacterial cell. And there's something very important here. No, remember I said I'm going to teach you everything you need to pass any exam under this aspect. Now, the cell wall of bacterial cell is made up of peptidoglycan. 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 Some proteins and some carbohydrates. Now, it's really very important you know this because this differentiates the cell wall of a bacterial cell from the cell wall of a plant cell. The cell of a plant cell is made of cellulose. Cellulose. And also, this also differentiates the cell wall of a bacterial cell from the cell wall of a fungi. The cell wall of a fungi is made of chitin, a polysaccharide, chitin. So please note all those things. Now, the next thing we're talking about now is the last layer inward, and that's the cell membrane. So every cell has this membrane, All cells have it. Now, so that's the last layer inward. So we call it the cell membrane. So this cell membrane has another name. So we can call it plasma membrane, or we call it plasmalemma. Plasmalemma. So it has another name like that. So cell membrane or plasmalemma. So those are the other names of it. Now, so it controls what goes into the cell and what comes out of the cell. That's the function of it. It controls what goes into the cell and what comes out of the cell. So all cells have these ones. Every cell has it. Now, the next one is the fluid-like material that is found inside the bacterial cell, and we call this cytoplasm. So cytoplasm. Now, this is like a fluid or gel-like material that found inside the cell, and its function is that it serves as a medium for chemical reactions to take place, or for kind of biochemical reactions to take place inside the cell. So that's the function of it. And also, that's where all the cell organelles are suspended. That's where all the cell organelles are suspended. Now, the next one, which is the last point I'm going to mention now, is the nucleoid, or let's say the DNA, and that's this one, this guy here. So we call it the nucleoid. Now, the nucleoid is like the region where the DNA of the bacterial cells are found, or let me say the region where the DNA of the prokaryotic cells are found. Because remember, I said bacterial cell is a prokaryotic cell, and its DNA is not found in the nucleus. Its DNA is not found in the because they do not have nucleus. Remember in the first place. So they do not have nucleus, they don't have mitochondria, they don't have chloroplast, they don't have vacuole, because those things are membrane bound organelles. So they don't have it because they are prokaryotes. Now, they don't have those things, but their DNA is found in the nucleus, not nucleus now, nucleus. So that's the region where the DNA of the prokaryotic cells are found outside the nucleus, not the nucleus now. Now, because they do not have nucleus. So we say the question you have to pay attention to, I saw it one of the exams, they asked that the chromosomes of kingdom monerans, are found. So it means that they are found suspended in the cytoplasm. All right, so now the next thing is that since prokaryotic cells they lack nucleus, so they have all their organelles suspended in this cytoplasm. All right, so see, this is the end of the video for this class. In my next class, I'm going to explain the blue-green algae and also move to the next kingdoms of living organism, the kingdom Protista. All right, so if you have missed my first video, so make sure you watch that before watching this one and watch this one before you watch the next one. All right, so